Biochemical Engineering. This lecture, as you saw, is titled Multiple Interacting Microbial Population Prey-Predator Models. And what we're going to talk about today is something related to growth, something that we had done before, but in a new perspective. If you remember, when we did the chapter on the microbial growth, what we talked about was the growth of a single bacteria or microbe or a fungus also. So what is the difference between what we're going to do today and what we did earlier? Earlier, as I said, we had worked or talked about just a single microbe or bacterial growth. We had, if you remember, talked about different substrates, inhibitions, and so on, but in all these cases, we just confined ourselves to single microbial growth. So if I can go back, to the old slide here of typical microbial growth in a batch reactor, this is how it looked like, if you remember, the cell number versus time, and there's a slight lag phase for the growth to start, and then there is exponential growth, and then there is a stationary phase, and then there is a decay. During the growth phase, the exponential part of the growth phase, only the growth of the bacteria occurs. During the stationary phase, what happens? The amount of bacteria that is growing or microbes that are growing or being incubated in the reactor equals the amount that is dying. So therefore, there is a stationary. So there is no overall growth, but there is the growth of bacteria, but which equals the death of bacteria. And then you have the final death phase, where the death rate far overcomes the growth rate. As a result, there is a decline in the cell number. And the reason I'm showing this to you is because this is important, and I'll draw some more curves for you today, and then we can compare how it looked as compared to the single microbial growth case. And the most important model that we talked about is, and we used it several times, if you remember, just as a recap, is a monod growth model, which was obtained or first derived by J. Monod in 1950, and it says that mu being the specific growth rate equals mu max, the maximum growth rate, times the substrate concentration divided by Ks plus S, S being the substrate concentration and Ks being the growth constant. And this is very similar to the Michaelis-Menten kinetics, so therefore mu almost has a Michaelis-Menten kinetics, and when in the case when substrate is present in a large amount, this mu almost goes to mu max, that is becomes a constant, that becomes independent of substrate, and the reaction becomes zeroth order. When substrate is very low, then it goes to a first order rate constant. So here again, this is a plot of mu versus the substrate S, and for low concentrations of the substrate, it almost behaves like a first order. For very large concentrations of the substrate, here it almost behaves like a zeroth order. And there are other kinds of growth models that we talked about, but we're not really interested in Malthusian and so on. We really hadn't been using these models. There is a modified monod growth model, which includes some more the rate of decrease at high values of initial concentration and different other models. But this is what we need to stick to and what we need to remember, the monod growth model, and we're going to use that today again. Now, apart from the very simple monod growth model, what else did we study? We studied the effects of mass transfer, which is valid here again, and we can apply these same techniques, and we can come back and talk about this at the end of this lecture. And we had also looked at multiple substrates here. So where two different substrates, S1 and S2, both are used by the growth, both are used for the growth of the bacteria or the microbe X. Okay, and then we had also talked about effect of inhibitory substrates that inhibit, but and allosteric inhibition also, which is the substrate reacting with the microbe to form X plus daughter cells, and that reacting again to form SS, that gives the daughter cell plus XS, that can again give X. So these are different kinds of inhibitions we had talked about, but what is important to note is that even in these inhibitions or this particular case of multiple substrate, we had always looked at a single microbe or single bacteria. What we're going to talk about today is multiple microbes of bacteria. Now, you might ask that what's different when you have multiple bacteria or multiple microbes? Well, nothing is very different, but something still is different. And what is not different is that both of these microbes of bacteria are going to use up the substrate. Now, you might ask that if that is the case, then both these microbes or bacteria may be working independently. They might be consuming nutrients independently and growing independently. Then where is the problem? Well, there is no problem if that is what happens. That is, both these microbes or bacteria grow independently. But we are going to consider the case when they do not work independently, or in other words, the growth of one affects the growth of other. And that particular kind of growth is termed as interacting growth. Okay, so that's why I call multiple interacting microbial population, and we'll give specific attention to the preparator model, and we'll redo some of the old stuff, but in a much more complicated light, and looking at it from a more complex perspective. Okay, so let's start working on it. So, as I said, this is multiple interacting microbial population. So, the stress is on both multiple and interacting. So, till now we discussed growth of single microorganisms. All right. So, now we are going to start discussing several different microorganisms which interact during the growth process. Example: biological wastewater treatment, cheese manufacture. These are examples from everyday life, where multiple bacteria or multiple microbes work together. Okay. It turns out that mixed populations of microorganisms are the rule rather than exception. Okay. And natural cycles, what is their role actually? Why are these multiple interacting growth models or multiple interacting bacteria important? So, natural cycles of carbon, hydrogen, nitrogen, oxygen require on our planet require active participation of many different microorganisms. Okay. So, what about examples? Let's talk about an example which is symbiotic example, where the growth of one bacteria helps the growth of the other bacteria. Okay. So, one is a microorganism and the other one is other organism, and the function, what is the role, how this happens. So, let us consider this example of a flagellated protozoa. Okay. This is the first example. Flagellated protozoa. This is termites, and the function is protozoa hydrolyze, the hydrolysis of cellulose for termites which the termites alone cannot digest. Okay. So, they cannot digest these cellulose molecules, and so the protozoa helps them hydrolyzing into glucose. Okay. And in return, what is done is that they turn the flagellates turn in host. They turn in as host, and they host the bacteria to provide. So, flagellates host bacteria and provide cellulase. Cellulase is an enzyme that helps in hydrolysis of cellulose. So, essentially this is what happens. So, the protozoa hydrolyze the cellulose for the termites. They are the ones which can hydrolyze the hydrolyze the cellulose, and because the termites cannot digest, and the flagellates host the bacteria and provide the cellulose. So, let us look at another example, same symbiotic, and in this case it is a symbiosis between algae and protozoa. Okay. So, what is the function? Function is that each protozoa holds around 50 to hundreds of algae. And what does algae do? It is well known what an algae does. What does it do? The algae use light, photosynthesis, to fix the carbon dioxide and free oxygen. Okay. So, that which the protozoa, this oxygen could be used, protozoa uses oxygen to oxidize nutrients and liberate CO2. Okay. So, these are the two examples we looked at. So, first one is the flagellated protozoa and termites, and second one is the algae and protozoa. So, as I said that each protozoa holds around 50 to 100 of these algae, and the algae essentially what it does, it uses light, photosynthesis, to convert to fix carbon dioxide and release oxygen, and this oxygen is taken up by the protozoa for its metabolic activities, and it releases carbon dioxide, which is then again taken up by the algae. So, there is a symbiosis between these. So, this is one set of interaction where the growth of one particular organism helps the growth of other particular organism. Okay. So, you can have different cases where the growth of one organism helps the growth of the other, that is symbiotic. Then you can have cases where they just work as hosts, but the growth of one does not necessarily, unlike in the algae protozoa case where the growth of one helps in the growth of the other. You can have cases where the growth of one does not necessarily help the growth of the other, but what it does is that it is sort of neutral. And then you can have competition and the Darwinian model, which is what we will look at in a few moments. So, the next thing that we look at is the predator model, and this is a more interesting model, because if there is no growth of one organism doesn't hamper the growth of the other, it's not as interesting. Here what happens, there is competition, and the survival of the fittest theory of Darwin comes into play. Okay. So, what essentially happens in a prey-predator model is that, let us consider a population, it is not even consider microorganism, let us consider a forest population, and you have grass which is eaten by the herbivores, let us say the cows and the cattle, and then you have let us say crocodiles and lions and tigers which feed on these herbivores. So, the carnivores which feed on this herbivore. So, you have the grass, the greens, and they are eaten by the herbivores, and the herbivores are eaten by the carnivores. Okay. So, this is a model for growth of this entire population. So, so essentially if I think of a forest population over here, you have the greens, which are eaten by the herbivores, and the herbivores are eaten by the carnivores. So, in this system, for example, as you can see, the greens are not eaten directly by the carnivores, but it is only through the herbivores. Now, if the greens are assumed, if you assume that the greens are in plenty, then the herbivores become the prey and the carnivores become the predator. Okay. Even if greens are not in plenty, you can still assume that the greens are the herbivores are the prey and the carnivores are the predator. But if they are in plenty, then it's easy to assume that one is the prey and the other is the predator. So, how is this model going to work? What will happen is that the herbivores eat on the carnivores. Now, the carnivore population, the ironic thing about it is that the carnivore population is dependent on the herbivore population, or in other words, the carnivores need to eat the herbivores in order to grow, but at the same time, if all the herbivores are depleted, then they have no access to the greens, the carbohydrates that are taken up by the herbivores. As a result, which will happen, this entire prey population system will fall apart. So, this prey population, the prey predator chain that we have, the prey predator cycle that we have, so to say, is a very important component of the food chain, the most important component of the food chain. And in order to keep this cycle alive, we need to keep in order to keep the food chain alive, we need to keep the cycle alive. Okay, that's one of the important points. So, how do we keep this cycle alive? That is what we are going to look at. So, let us look at the case of two microorganisms which are part of the prey predator model in a way. So, one is E. coli and the other one is Staphylococcus aureus. Okay. Now, experiments, if you do experiments with these systems, this is what you will see. So, let us see. This is my and this is E. coli, and let us see, this is Staphylococcus aureus, and this is Staphylococcus aureus mixture. Okay, I will explain everything. So, this is the number of cells per unit volume, this is time. Okay. These two are E. coli, okay, and these two are Staphylococcus aureus. The red line is for mixture, blue line is for alone for E. coli. Similarly, black line is for alone for Staphylococcus aureus, and green line is for mixture. So, if you look at this curve, what is it that you find? What you find is that when Staphylococcus aureus is allowed to grow alone, this is its growth rate, which is pretty high. Now, when you allow it to grow in a mixture with E. coli, its growth rate decreases substantially. Now, look at E. coli, it's more or less the same, but still when you allow it to grow in mixture, it grows slightly faster than Staphylococcus aureus. Okay. So, why does that happen? Let us try and understand why that happens. The reason why it happens is because E. coli over here that you see has a much higher growth rate than Staphylococcus aureus, and the substrate that is available, it's available to both. So, E. coli would eat up all substrate or much of the substrate that's available, and as a result Staphylococcus aureus cannot grow. So, this is a case where the growth of one microorganism, in this case E. coli, hampers the growth of the other one, Staphylococcus aureus. So, these are interacting in that way. Now, what we want to study is, now what we need to do is come up with a model of these kind of prey predator models. So, I want to give you another example. This is one example. The other example that I want to give you is this: a protozoa versus a bacteria. Okay. So, silates, this is a protozoa, its name is Tetrahymena pyriformis, okay, and the bacteria is aerogenes. Okay, this is a bacteria, and the y and the y axis is essentially number per unit volume. Okay, the x axis is time. So, we are trying to grow both of these together in the same medium, let us say, and if I am to draw this one with black, the protozoa, if I am to draw this with black, this is how it looks like. It goes up, down, and up. If I am to draw the bacteria, this is how it looks like. Okay. So, this is the green is this one and black is this one. So, what do you notice over here? What you notice is that whenever the protozoa is up, the concentration of the protozoa is up, the concentration of the bacteria is low. Whenever the concentration of the bacteria is up, the concentration of the protozoa is low. So, they are going through some sort of a cycle where the concentration of these two together is more or less a constant, but each when one goes up, the other goes down. Okay. And this is known as prey predator oscillations. Why does this happen? Let us try and understand this qualitatively, why this happens. So, let us say that the one is a prey and the other one is a predator. So, when the predator population goes up, it goes up because it is consuming the prey. As a result, the prey population has to go down when the predator population goes up. Now, what happens as soon as the predator population, prey population goes down, there is less food for the predator now, and as a result, the predator starts to die. And the predator reaches a point when the predator starts to die and decrease in number, the prey population goes up because there is not enough predator that is eating up the prey at this point of time. Now, when the prey population goes up, then the predator starts to eat up the prey again, and then it goes up again, and as a result of the eating up, the prey population goes down, and this cycle continues again and again. So, let us try and understand how to model this entire system. So, the model that we talk about is known as a Lotka. The model was derived by these two scientists, Lotka-Volterra model. So, it is called the Lotka-Volterra model of prey predator oscillations. So, let us say N1 is a prey and N2 is a predator. So, if I am to write a balance for N1, a simple ordinary differential balance for N1, so I would write ddt of N1 equals A N1. This is the growth of prey from the substrate that is there, could be greens or whatever it is eating, but remember the prey growth is not dependent on the predator. Prey is growing by itself, and the death of the prey, the only way, so that is where the negative sign, because this is the growth, so there is a positive sign, is a predator, this is a death, so therefore it comes with a negative sign, and that is given by gamma some constant times N1. Why N1? Because more the number of prey, the possibility of more prey dying is of course more, and therefore it goes as N1 times N2. This one depends on the predator. Why? Because this is death from consumption by predator. Okay. So, therefore, you have the growth here and the death here, and so this is the first part, that is the balance on the prey. Now, if I am to do a balance on the predator, that would be ddt of N2 equals minus Bn2, which is death of predator. Why is it independent of the prey? Because the death of the predator has nothing to do with the prey in general. It has to do with the fact that predators are dying naturally, plus the birth of predator. Remember, we had the death of the death of prey from consumption by predator. Now, the rate at which prey is dying should be similar or proportional to the rate at which predator is growing, because this food, the prey is being consumed, and that food essentially gives rise to the predator, that gives to the growth of the predator. Okay. So, this is therefore proportional to gamma times N1 N2, which is the death rate. But is it equal to the death rate? No, it is not equal. Let us multiply this by a factor epsilon, because not all the food that has been consumed by the predator goes in the formation of the predator itself. So, a part of it. So, epsilon is typically less than 1. So, growth of predator through consumption of prey. Now, if I look at these two equations together, okay, probably I will write them down again one after another, so that it is easy. So, first one is dn1 dt equals an1 minus gamma n1 n2, and the second one is ddt of n2 equals minus b n2 plus epsilon gamma n1 n2. So, if I look at these two equations, this is my model, prey predator model. If I look at these two equations together, what do I see? What I see is the fact that I can get a steady state out of it. So, it is possible to have a steady state. So, if I equate the left hand side to 0, then the first equation would give me N2 equals A over gamma, that is it, N2S, and the second equation would give me N1S equals B over epsilon gamma. Okay. So, these are my steady states. Okay. Now, the next step that I want to do is I want to convert these two equations into dimensionless form using the steady state values, or in other words, let us call y1 equals n1 over n1s and y2 equals n2 over n2s. Okay. So, this is what I am going to do. So, when I do that, the dimensionless equation that I get is given by dy1 dt equals a 1 minus y2 y1 and dy2 dt equals minus b 1 minus y1 y2. So, how do I solve these equations? If you look at these two equations, both are functions of time, and each of them involves a coupled. So, each of them are nonlinear, coupled, nonlinear, coupled ODEs. Okay. Can I solve them as it is? The answer is no, I cannot solve them as it is, but I can solve them in what is known as solution possible in phase space. Okay. So, not in the space of the variable t, not in the time domain, but in the phase space, that is y1 versus y2 space. So, y1 versus y2 space. So, that is where solution is possible. How do I do that? All I need to do is divide equation, let us say this is a and this is b, divide equation a over b, and you get dy1 dy2 equals a 1 minus y2 y1 divided by b y1 minus 1 y2. So, this is what I get. Now, can I solve this equation, the one that I have over here that I have boxed out? The answer is yes, simply by transposing the terms that contain y1 to this side and the terms that contain y2 to the other side. Let us do that, and when we do that, I get b y1 minus 1 over y1 dy1 equals a 1 minus y2 over y2 dy2. Okay. So, this is what I get, and so I can go ahead and solve it. Let us do that. 1 minus y1 over y1 dy1 equals a 1 over y2 minus 1 dy2. So, if I integrate this on integration, I get b y1 minus ln y1 equals a ln y2 minus y plus c. C is a constant over here. So, now I can convert this into exponentials. So, y1 to the power y1 over e to the power e y1. So, this comes here and this becomes e to the power e y1 whole to the power b times y2 over e to the power y2 whole to the power a equals e to the power c. So, this is my solution of this equation, but solution in phase space. Okay. So, it is possible to solve this, but it is only possible to solve this in the phase space. Okay. So, when I solve this, I get this equation and solve this in the phase space. What kind of plot do I get in the phase space? I can convert this back into my dimensional form, because y1 and y2, I know them, how they vary in the dimensional system. So, simply write n1 over n1s divided by e to the power n1 over n1s whole to the b, and this is n2 over n2s divided by e to the power n2 over n2s whole to the power a equals e to the power c. Okay. So, what is the plot in the phase space? How does the plot look? This is how it looks. Let us draw this. This is a phase space, remember. So, there is no time here. One axis is n2, the other axis is n1. So, let us plot let us say this is my center. Okay. This is my center, and this is n2s, the center is, or the point steady state. So, n2s and n1s. So, this is my steady state. Now, how would the plot look? The plot would look something like this. What are these? These are sort of limit cycles, so they are going in cycles again and again. And if I am to go back to one of the earlier plots over here, the prey-predator oscillation, see, this was how it looked in the variable space. If I am to plot n1 and n2 over time, this is how it would look like. And if I am to plot them against each other, so look at this, what happens is when this is maximum, this is minimum. Or in other words, if you look at it from the phase space, this is how it would look. So, this is large. If this is n2 is large, n1 is small. Okay. Or if n2 is large, here it could be large, but if this is a part that forms a part of the limit cycle. So, it goes on and on in limit cycles. Okay. Now, so this is a circular thing in the phase space that happens. Now, what I have to figure out is this thing that you are looking at here, is it stable? Is it stable? And that is a very important question that we need to answer. Why do we need to look at the stability of prey-predator models in the phase space? The reason we need to look at the stability of prey-predator models is because, as I said, that we want the food chain to be stable. If the prey-predator model falls apart, then the whole ecosystem will fall apart. The food chain is a natural way that has, through evolution, that has happened to preserve the ecosystem as it exists today. Now, if the food chain falls apart, then there is problem, and this is not just confined to bacteria and microorganisms that we are dealing with, but also the general food chain, and that's why I gave these examples of the carnivores and the herbivores and the greens to start with. Okay, so what is necessary? It is necessary that this dynamic steady state that is produced is sort of stable. I mean, the reason I call it a dynamic steady state is that, you see, it goes through these cycles. If it's a very oxymoronic way of calling it the dynamic steady state, but still, I mean, you have a steady state, but the dynamics of it goes around the steady state, just as I showed. You have the steady state at the center, and the cycles go around it, so the dynamics of it, in the phase space at least, go around it. And that's why there's a periodicity involved. We talked about how the waveform, you know, I showed you the waveform of the prey and the predator, so there's a periodicity involved, and there is, so it's, you can sort of call it like a periodic steady state or a dynamic steady state. Now, we need to understand whether this dynamics that is there, the dynamic steady state, the periodic steady state that is obtained, is it stable or not? Because if it is unstable, then the whole food chain will collapse, not just because of the microorganism thing, but in general, and we are in trouble. So, we have to look at and investigate the stability of this system. We have looked at stability of problems, you know, we had looked in great detail about stability of reactors, and what is the, you know, we had done step by step, so you can go back and look at those lectures. I will quickly summarize. What you need to do is, when you look at the stability of a dynamic state, essentially you need to take the dynamic equation, linearize it, get the Jacobian, and then look at the Eigen values of the Jacobian. So, what was the necessary condition for the system to be stable? That the Eigen values have to have a negative real part. So, what were the different points we looked at, and different cases we looked at? One, when the eigenvalue had a positive real part and a negative imaginary part, or imaginary part as such. So, the system will be unstable and it will have oscillations. The other possibility is that the system will have a negative real part and an imaginary part, in which case it will have oscillations, but it will be stable. And then you can have a zero real part and an imaginary part, in which case it will be neutrally stable, but it will have oscillations. You can have a real part, a positive real part with no imaginary part, in which case it's completely unstable, and you can have with no oscillations, and then you can have a negative real part with a zero imaginary part, in which case it is going to be stable and without oscillations, and you can have a zero real part and a zero imaginary part, in which case it is going to be neutrally stable with no oscillations. Now, as you can almost predict in this case, that what is going to happen, is it going to have a real part positive or negative? We have to figure out. But what about the imaginary part? You can almost predict in this case that the imaginary part is going to be there. Why is that? Because just as I showed you, the case, you know, the diagram over here, the prey predator model here, as you can see, it goes through these oscillations. Okay, so there is no way it can go through these oscillations unless the imaginary part is there. So, there has to be an imaginary part that we can almost predict. What we have to figure out is the real part positive or negative, or is the real part zero? So, at long, you know, when you look at it at long times, are these oscillations, they are going to be there, but are these oscillations going to slowly decrease in amplitude and then, you know, become stable? The system is going to become stable. So, that is what we need to look at. So, for that, what we need to have is that we need to go back to this system of equations. So, this is my system of equations, and this is equation 1, let us call this F1, and this is F2. What we need to look at is, we need to look at the Jacobian. So, if you have forgotten, I will recap how the Jacobian. So, A is the Jacobian, that equals del F1, del Y1, del F2, del Y2, del F1, del Y2, del F2, del Y1, and del F2, del Y2. Okay. So, this is my Jacobian. I write it one more time. So, A equals Jacobian equals del F1 del Y1 del F1 del Y2 del F2 del Y1 del F2, del Y2. Okay. So, now for our case, which is this, these are my F1, F2, I need to find what my Jacobians are. So, let us go ahead and do this now. So, A would be, the first term is del F1, del Y1, and that is simply A times A times 1 minus Y2. Okay. Then next term is minus, so which is del F1 del Y2, which is minus A Y1. Now, del F2 del Y1, that is B Y2, and del F2 del Y2 is minus B 1 minus Y1. Now, I have to find out what my eigenvalues are. How do I find that out? I have to first calculate the determinant of A minus lambda I and equate that to 0. This will give me my characteristic equation for eigenvalues. Okay. So, let us form the characteristic equation first. So, A minus lambda I would be equal to A 1 minus Y2 minus lambda minus A Y1 B Y2 B Y1 minus 1 minus lambda, and the determinant of A minus lambda I equals equals lambda minus A 1 minus Y2 times B 1 minus Y1 plus lambda plus AB Y1 Y2 equals 0. Okay. So, the next step is lambda minus A 1 minus Y2 lambda plus B 1 minus Y1 plus AB Y1 Y2 equals 0. Okay. Now, so the characteristic equation is formed as lambda square minus A 1 minus Y2 lambda plus B 1 minus Y1 lambda minus AB 1 minus Y2 1 minus Y1 plus AB Y1 Y2 equals 0. That is my characteristic equation. When I simplify it, I get lambda square minus A 1 minus Y2 lambda plus B 1 minus Y1 lambda minus AB 1 minus Y1 minus Y2 equals 0. Now, I have to evaluate the lambdas at the steady state. So, steady state in my case means Y1s equals Y2s equals 1. Why? Because Y1 was defined as N1 over N1s and Y2 was defined as N2 over N2s, right. So, obviously, at steady state these values are going to be 1. So, if I put 1 over here, this term vanishes, this term vanishes, and what I have is lambda square plus AB equals 0, or in other words lambda equals I imaginary times AB. So, this is my answer, plus minus, sorry. So, this is my answer. This is what I find, that plus minus. So, two values of lambda that I get are plus and minus I. This is the conjugate roots I square root of AB, which means that the real part of lambda, which means that the real part of lambda equals 0 and imaginary part of lambda equals plus minus square root of AB, which means what? That the system, so if real part of lambda is 0 and imaginary part of lambda equals plus minus square root of AB, this means what? This means that the system is neutrally stable with oscillations. So, the oscillations part we had already seen, known, and we had all sort of predicted, and what we find over here, the new thing that we find over here is that the system is neutrally stable. It is not asymptotically stable, it is not unstable, but it is neutrally stable. So, it just remains as it is, and that is why the pictures that we drew over here, this one for example, and this one. So, this one, these oscillations will continue, and in the phase space also it will keep going on in these circles. Now, you can ask me, what are these different circles about? Why do you have so many different circles? It depends on the initial condition. Remember, these are dynamic equations we are solving. So, for one set of initial condition will be on this trajectory, another set we could be somewhere here, and if you keep changing the initial condition, you could go further away from the steady state. But remember, these are dynamic systems, so they all depend on the initial condition. So, these circles are for a particular initial condition, but for a given initial condition, the system will continue going on this cycle forever. It is not going to come in, and it is not going to happen that it is going to come in like this. That is not going to happen. It is going to go in on and on these cycles. The reason being that the real part being 0, the system being neutrally stable. Okay. Now, what we are going to do next is competition and selection. So, we are looking at competition here also, but this is, remember, what we did here over here is the predator model in general, but our interest is essentially the chemostat. So, what we are going to do next is, and very quickly, is competition and selection in a chemostat with limited substrate. Substrate is not unlimited, so it is a limited substrate. So, what does it mean? What does it mean when I say that substrate is limited? What it means that in addition to the prey and the predator, I need to write a balance now for the substrate, which we will do. So, let us say one is a prey, the other one is a predator, and N1 again is a prey and N2 is a predator as before. So, ds dt, which is the substrate, is, if you remember, we wrote this chemostat equation before, dilution rate d times S naught, the initial amount of substrate present, minus S, minus 1 over ys mu S S times N1. So, mu S is the specific growth rate of N1. So, what is happening? Substrate is coming in and going out, and the prey here, which is exactly, I gave you the example of the herbivores and the carnivores and the greens. So, think of the substrate as the greens, and the prey is eating on the substrate and growing. As a result, the substrate is being depleted, and this is leading to the growth of the prey. So, the prey N1 is growing, and this is proportional to the growth rate, the consumption of the substrate, and because of this growth process, the substrate is being consumed. And how much is the substrate being consumed? That depends on the specific growth rate. So, larger is mu, more substrate is being consumed. Okay. So, larger is mu, more substrate is being consumed over here. Okay. Is that clear? So, the next thing that we do over here is write the balance for the prey. So, this is the substrate and this is the prey. So, that equals minus d N1. This is entering the system because of the because dilution rate, or in other words, the entry of the exiting the system and entry. So, essentially this is essentially the exit term, plus mu S time of function as a function of S times N1. So, this is the growth of the prey from the substrate. It is eating on the substrate and growing, and this is the growth rate, and that is why it comes with a plus sign. This one comes with a negative sign because it is exiting the system. And then you have minus 1 over yp mu p N1 times N2. What is this? This comes with a negative sign. I will put the negative sign here actually. This comes with a negative sign, which means that this is not the growth, not the birth, but the death of the prey, and this is happening because the predator N2 is eating on the prey. And at what rate is it eating on the prey? At the specific rate. This is not a growth rate anymore, it is like a death rate, but you can still call it a growth rate, which is mu p as a function of N1, and yp is a selectivity ratio. And then we have to write the model for the predator. I hope you can see it here. This is the model for the predator. Let me write this in green. So, dN2 dt equals minus dN2, that is, why is there no entrance? See, for the substrate there is an entry term ds naught, but for the prey and the predator there is no entry term, because no prey or no predator is entering the system, only the substrate is entering and it is growing. Prey and predator are growing, and this is given by plus mu p N1, mu p as a function of N1 times N2. Okay. So, this is the overall model that we have. Now, what we need to compute in the next few minutes is the steady state. So, there are 3 steady states for the system. Steady state 1 is N2s. If you go and solve it, you will find this, and the solution is not very difficult, equals 0, N1s equals 0 and S equals S naught. Steady state 2 is N2s equals 0, D equals mu s of S, and which means that D and mu s of S is given as mu s max times S over Ks plus S, and mu p of N1 is given as mu p max N1 divided by Kp plus N1. Okay. So, this is 1. So, from this you get steady state, I am still on steady states 2. So, this gives you mu ds as this gives you Ss as Ks d over mu s max minus d, and N1s equals S naught minus Ss times Ys. And let me write steady state 3 quickly. Steady state 3 is what I get are N1s equals Kpd mu max minus d, and N2s equals N1s Yp mu S minus d over d, and S itself comes out to be minus b plus, there plus minus, but I will take only one of the roots because the other root does not make sense, 4 S naught Ks, this is half, where b equals 1 over Ys times d times Ks minus S naught plus mu max N1s. So, this is a little complicated solution, the steady state 3. Steady state 1 and 2 are very easy. Now, what you can do is, as we did in the previous cases, go and look at the stability of these steady states. How do you do that? You just form the Jacobian like we did before, and you have a 3 by 3 Jacobian, and you figure out how the eigenvalues look like, and the positive, if it is a positive real part, then you know that the eigenvalues are the system is unstable. If it is a negative real part, then you know that the system is stable. If it is 0 like we had before, then you know more or less that the system is neutrally stable. So, this sort of concludes what we are trying to do over here, but what I will do in the next couple of minutes is just run through what we had done in the previous lectures, in all these 24 lectures put together that we had been doing. So, we started with initially there was an introduction given on the fundamentals of biology and so on, and then to enzymes, and we started with the biochemistry and thermodynamics of enzymes. We looked at different kinds of inhibition, competitive, non-competitive, substrate inhibition, different kinds of inhibition. We looked at the effects of pH and temperature, and then we moved on to immobilized enzymes. So, in mobilized enzymes there were no mass transfer effects, and we moved on to immobilized enzymes. And in immobilized enzymes, we found that there was mass transfer effects that were there, that were hampering the growth of or the reaction kinetics of the enzymes. And so, we looked at the effect of mass transfer in immobilized enzymes and the effectiveness factor in immobilized enzymes. And we looked at how kinetics is being hampered, or in other words, how the reaction rate is being decreased because of the presence of immobilized enzymes and the mass transfer rates there. And then the next thing we looked at was microbial growth, the different phases of microbial growth, the models for microbial growth, and effects of mass transfer on microbial growth, and so on. And we also looked at the effects of multiple substrates and inhibition. And today's lecture we looked at the effects of not multiple substrate, but multiple microorganisms and the interaction between them. And there is a continuity between what we did. And then we looked at bioreactors and design of bioreactors and stability of bioreactors. And just today also we looked at what happens when so we had looked before at what happens when a microorganism grows in the bioreactor and the stability of the growth process. Today we looked at the stability of interacting growth processes. So if two microorganisms are growing together, then how does this interaction relate to the growth process? Does it hamper the growth of one? Does it accelerate the growth of the other? And so on. Then we had moved on to a different area altogether and looked at receptor ligand binding. We had looked at in great details at the kinetics of receptor ligand binding. We had looked at a particular disease called familial hypercholesterolemia, and we had tried to understand how the receptor ligand binding, the kinetics of this, influences this disease. And we had studied in significant details the process of receptor mediated endocytosis, which is a very fundamental and important process in many physiological systems, and ligand receptor binding plays a very important role. So this is what we had been doing with a very sort of a quantitative approach at this part of the course, and the other part of the course deals with a more qualitative approach. I hope you enjoyed the process and had learned something in the entire process of how to deal with biochemistry and biology, and bring in a chemical engineering perspective of transport and reaction into it, and make it more quantitative than we typically do. So thank you.